Hey A&Pers, this is Dr. Jones checking in. This is A&P II Lab 2 Video 1, and we're talking about blood in the cardiovascular system. So, starting at the top of page 12, let's talk about blood cells. So the red blood cells, all these little red discs, the name of this shape is called a biconcave disc because it's concave, it bumps in on both sides. So it's a biconcave disc. These are red blood cells, and the technical term for a red blood cell is an erythrocyte, and they're about half of your blood. So the fluid portion of the blood is going to be plasma, and all the solid bits together, all of these cells, are going to be formed elements. Of the formed elements, the erythrocytes, the red blood cells, make up about 99.9%. So they end up being about half your blood, depending on your hematocrit. So the rest of these cells on this model are going to be different types of white blood cells. The general term for white blood cells are leukocytes, Leukocytes, leukocytes. And there are two types of leukocytes. First, we'll talk about the granular leukocytes. First, let's talk about the neutrophils. So you see these pink ones with the purple. Okay, so the nucleus here, these are also called polymorphonuclear leukocytes because there are lots of lobes to the nucleus and they have a weird shape. So polymorpho means shape, poly means many, polymorphonuclear leukocytes. Okay, and they can have as few as 2 lobes to the nucleus and still be a neutrophil. Neutrophils are the most numerous of all the white blood cells at 50 to 70%. They do your infection control, and they're the first to show up at a wound, and they make up part of the pus. The next blood cell on our list is the eosinophil, and it takes up the eosin stain in the granules, and so it looks more orange. And it also has a bilobed nucleus. Sometimes on the slides, it's hard to tell a neutrophil from an eosinophil. The neutrophils are far more numerous, though, at 50 to 70% of your white blood cells. Your eosinophil is only 2 to 4%. So if you're looking at a slide and you see 2 that look identical on the same slide, that's probably neutrophils. That's probably not eosinophils because there's just not that many. The next on the list is the basophil, and that'll be this dark one in the corner. It takes up a dark blue stain and in the granules. And on some of the slides, you can't even see the nucleus because the granules are stained so darkly. These are the least numerous of the white blood cells at less than 1% of your white blood cells, and they cause inflammation. Yeah, thanks. So all of the granulocytes, the neutrophils, the eosinophils, and the basophil all end in phil. So Phil named all of these after himself. I'm joking, but these are the granulocytes. The agranulocytes are the monocytes and the lymphocytes. The monocytes are these great big ones, and they have kind of a bent, depending on the angle you see it, it might look kind of round, it might look bent like kind of a kidney bean, and these are the monocytes, and then the lymphocytes are the only ones that do your specific immunity. The rest of these white blood cells all do non-specific immunity, where they just go to an infection and kill it. There are different types of lymphocytes, the T cells, the B cells, and the natural killer cells, and we can see kind of different kinds of lymphocytes here. The monocytes are the biggest, but some of these lymphocytes are pretty small. And on the slide, compare them to the size of a red blood cell that's near them. And then these tiny specks, these little blue bits in between, are going to be your platelets. The platelets are not cells, they're cell fragments, and they function in clotting. So do we say what is the function of a red blood cell? To carry oxygen and carbon dioxide. Okay, and then the 2 agranular types of cells, the monocytes and lymphocytes, both end in the root site. They do have granules, but when we named them, we couldn't see them, and they don't show up on a light microscope. And they're actually, the granulocytes are made through a different pathway, so it is still a useful thing. Okay, so let's switch to the blood typing. I'm going to move this model out of the way. Okay, so let's talk about blood types really quickly. This is my version of the picture in the lecture and lab book. Looks like on one version of the book, it's figure 34.4 on page 463. That's probably one of the lab books. So type A has these A surface antigen, which our book shows as circles. Type B has a B surface antigen. Type AB blood has both A and B surface antigens. And type O blood, which originally meant 0, but it got misinterpreted at one point to an O, has not the A or the B surface antigens. So if you have type A blood, in the blood you have anti-B antibodies. Now, if you mix the anti-B antibodies with type B blood, it will attack and agglutinate. Do I have that word written down for you? I don't. Agglutinate. L-U-T-I-N. It will agglutinate, which means to clump. And then that causes hemolysis. Hemo for blood, lysis for a loosening of, which in this case is sort of an explosion. So if you're born with type A blood, you're born with anti-B antibodies, so that if any type B blood gets in your system, these anti-B antibodies will clump with them, cause agglutinations, and then rip those cells apart and destroy them, hemolysis. So if you're born with type B blood, you're born with anti-A antibodies. So they're anti-A. They attack the A. If they find any A, they agglutinate and then rip those cells apart. Type AB blood does not have these antibodies because they would die. Okay. And type O blood has both. Okay, so the way we test, let me walk you through this. Zoom in and walk you through this. Is we make a well anti-A fluid and anti-B, and then we put a sample of blood in it. Yeah, I should get that on screen, sorry. So if we have a reaction in the anti-A, that means that the anti-A antibodies found a surface antigen. So this blood sample had A in it. So let's say the same sample, we see no agglutination, no clumping, no stippling, no reaction in the anti-B. The anti-B only attacks B. So this blood sample did not have a B. Okay, let's say our next sample has a no reaction in the A. So the anti-A antibody found no A surface antigen. I should have warned you, like everything in this section starts with A. I apologize, it's kind of a pain. But let's say for this sample of blood that the anti-B antibodies found some B surface antigen, which is the only thing they attack, and when they attack, it causes these clumps, these agglutinations, the stippling, the dots. Okay, so we've got no A reaction, so there's no A surface antigen here, but we did get a big reaction with B. So we have a B, type B blood. So what if we get agglutination in both the anti-A and the anti-B columns? Well, the anti-A only attacks A, so this blood has A surface antigen. And the anti-B only attacks B surface antigen, so there is B in this blood sample, so this is AB blood. We're just going to do all of them. So what if you get no reaction? The anti-A found no A, no A. The anti-B found no B, then you got 0. You've got an O blood type. But that's not, and this is called the ABO system. And in the ABO system, you can have type A blood, type B blood, type AB blood, or type O blood. But the ABO system isn't the only way we tell blood types. We also have the RH, and the RH stands for rhesus because they first found this in rhesus monkeys and then got curious and found it in human blood too. And with this rhesus factor, with this surface antigen, you either have it and you're RH positive, or you don't have it and you're RH negative. So the test, the serum that they use here is anti-D. So let's add our third well, and let's say that this blood sample that we already said was a type A reacted with the anti-D. The anti-D only reacts with the RH positive, only reacts with that rhesus surface antigen. So this blood sample is A positive. Okay, let's make another blood sample. Let's say we've got stippling in the anti-A serum, no stippling in the anti-B serum, and no stippling in the anti-D serum. So we know we have A surface antigen, we know we don't have B surface antigen, and we know we don't have an RH positive. So this is an A negative blood type. Yeah, we're just going to do it. So B with stippling is a B positive, which is always good life advice. And then let's do a no reaction, a stippling, and a no reaction, so that we've got no A, but we do have a B reaction, and we have no RH factor. So that's a B negative, which is bad life advice. And then let's do all stipples, all reactions, all the way across the board, AB positive. What if we have stippling in the first two, but no reaction in the third? Well, that's our AB negative blood type. And that makes O positive. So the only blood type left is no reaction across the board is O negative. And that's your universal donor because they don't have surface antigens to activate anybody's antibodies. So how do we test that? Well, usually it looks like these. I don't have these in any order, so let's just go through them. So here, I've got it upside down. Oh, they're all upside down. Okay. Flip. Okay. So here you can see this well has an A, so this is our anti-A antibodies. Here's a B, this is our anti-B antibodies, and here's our RH, so this is our anti-RH antibodies. So we have no reaction, so this is type O negative. The A did not find anything, anti-A found no A, the anti-B found no B, but the anti-D did find, so this is an O positive. All reaction, AB positive. So we've got anti-A found A surface antigen to react against, but nothing else did, so that makes A negative. Reaction in the A, no reaction in the B, reaction in the RH. What blood type is that? A positive. I think we already did this one. Still a negative. What's this? B negative. A positive. And O positive, because the anti-A found no A surface antigen, the anti-B found no B surface antigen, but the anti-RH did find the RH factor, so that's RH positive, so that's O positive. So that's all about blood types. Thanks as always for tuning in. This has been A&P II Lab 2 Video 1 about blood. In the next video, we will start in on the heart model. The education must go on. Dr. J out.